Our next speaker comes to us from the Joint Research Center, a European Commission in Italy, and has been very active in systems toxicology over the years. The title of Dr. Elisabet Berggren's presentation is In Vitro Data and In Silico Models for Predictive Toxicology. Please.
Okay, thank you very much, and I'm very honored to be here today, and it's a privilege, of course, to speak for this audience, and it's a privilege also to speak after the previous speakers that very well set the scene and helped me a lot in my own presentation. So thank you for being here. I am going to tell you about the SORAT project that is sorry? I have to be closer to the microphone. Do you hear me? You didn't hear what I said before. But that was, you can live without that. Okay, so the SORAT one is a project. It is one of the FP7 projects that is co-financed also by EU with the Cosmetics Europe. It's a very large project. It's a 50 million investment. And it's a cluster of projects. So it's a little bit different from most of these consortia that is formed under the FP7 or H2020. We are 70 research partners, and they are spread out in 16 countries. And it's 6 years, which is not really true. It's about to finish now. The projects are actually working for 5 years, and it's only the coordination action where I am active as well that is working in the last 6th year to try to package the results and feed it into future projects. This is the SORAT1 roadmap. As you can see, it became quite complex. We tried to make a sort of Gantt chart from the beginning, how things were feeding into each other. But it was not so easy with the different projects. So we had the 7 projects in some way feeding into a mainstream cluster. We created different working groups to create a cross-cluster environment to discuss certain issues. And at the end, I mean, we managed in some way partly to handle this. And just now we are here. We are quite close to the end. And the little book there on the mainstream of SORAT1 is our 5th annual report that just was published. The SORAT strategy is based on the toxicological mode of action framework to say actually how a substance adversely can affect the human health. And we are looking at repeated dose toxicity, and of course we are looking only on alternative methods and how we can build up a safety assessment. And we have a lot of scientific tools in our luggage from the different projects. So we have one project working on bioreactors, for example. We have models to link the in vitro and in vivo with biokinetics. We have the Cosmos project looking at different computational models. And we have the different cell models, multi-scale models from organ toxicity. And we have our cell factory producing new cell models to use in our testing strategies. And we, of course, also have a tox bank that is storage of all the data. And we are also based, I mean, on the sort of AOP concept, and now you know very well everything about the AOP concept. Thank you for that, Ellen. So I will not tell you so much about this. But of course, we were also sort of trying to look at the molecularly initiated event, individuate the key events, and look at if we could predict adverse outcome. We were sort of establishing some proof of concept of the SORAT1 project, something that is coming out on the cluster level. And the first one was actually to make adverse outcome pathways based on the current knowledge that we could collect from all literature. And it was quite a large work we did on this. And we were focusing on liver toxicity and liver pathways. Then the second level was actually that we were making integrated systems of the methods available within the cluster, and then try to predict certain toxicity endpoints. And the last level of the proof of concept is the application. It's actually to make safety, regulatory acceptable safety assessment based on our tools and the predictive toxicity. I just want to say also that the AUPs that were created on the level 1 of the proof of concepts are today available in the AUP wiki. That is collaboration between the European Commission, the Joint Research Center, and the EPA. And it's already available, and you can look it up and look at this tool where you can actually then put in and create your AUPs. Together, work them out and improve them. So the ToxBank wiki, we also selected the chemicals to test based on the idea of mode of action. So we did look at similar mode of actions to try to then benchmark our systems. So I just give you a flavor of some of the tools. This is a very nice system to show fibrosis. It's a co-cultured 3D system where you have HepRGs together with hepatic stellate cells. It was developed by Hemobio, the Hemobio project, and actually they are studying this during 21 days, and you can see how the collagen is sort of accumulating, and that is a pre-state for the fibrosis. And this is actually one of the models also we are using for the proof of concept on the level 3. Of course, we had the Screen Tox project working on human pluripotent, induced pluripotent stem cells, and they have managed to make hepatocyte-like cells that have been used then in repeated dose tox testing. And in the detective project, that is the biomarker project, and they have, for example, made this sort of reporter platform that you can see a lot of different readouts at the same time to provide response on cellular stress. And the NoTox project is very interesting. It's also using HepRG, but in 3-dimensional systems, and they connect it directly to biokinetic modeling that is sort of giving another sense to your cellular system. All the data is collected in the data warehouse of ToxBank, and it will be publicly, you can connect and you can see what is publicly available already. Some of the data is still protected due to that people are finalizing, but a lot of data is publicly available. And there was the Cosmos project, the computational model project. They have provided this Cosmos database that is open access. You can connect to it straight away. And you have a lot of high quality cured toxicity data available. And it's specializing, of course, on substances with cosmetics, of interest for cosmetics. And there is also this Cosmos 9 web portal that is quite interesting, sort of providing more computational tools and also looking at kinetics that you can actually connect to, and it's easy to use, and it explained how you can use it. In 2013, we invited some of our colleagues from the other side of the ocean to a meeting with Toxide 21 and Surat 1 to establish a collaboration, and it was very fruitful, and we have continued to work together, and we have a lot of connection to our Tox 21 and also ToxCast partners. And then, of course, as I said from the beginning, we want the scientific tools to sort of provide us with something that can give actually solution to safety assessment. And we have developed this sort of conceptual framework for safety assessment. And it's very much based on the idea that you collect all data that is available, you look at your exposure, what sort of rules of exposure you can be of concern, and so on. When you have collected everything you can know about this chemical, you go and look at what type of toxicity you believe it can cause in your body, if it's general toxicity, if it is target organ specific adversity you are looking at. You are looking if it can reach the organs, on which time scale and so on, and in which concentrations. You look at, and then in what is happening in the cell, on the toxicodynamics and the mode of actions, and you then at the end collect it all and try to make some sort of assessment of this. And perhaps at this stage of life and where we are with the science today, it might be that you end up in some sort of structured logic, but you actually can identify, if you don't make a safety assessment, you can identify the weak points. And that is something that is helping us to construct a future where we can use the network or the sort of workflow. We have 3 different case studies on this level. One is on the TTC that is provided on the Cosmos project, that is thresholds of toxicological concern, that is very useful for certain chemicals and products. We have also a read-across case study that I will mention a little bit more, and the ab initio one, that is the one that you actually assume that you don't have any sort of data at all, which we are struggling a little bit with. And then we have the read-across case study. We have 4 different scenarios, and we agreed on this in an, oh no, that, okay, that is that. I go back. So we agreed that at the workshop, we also invited experts from outside the SURAT one to sort of get a complete view of the read-across problem. So we have 4 different scenarios. The first one is chemicals that is sort of acting at the target without having any sort of metabolism. The second is where you have the metabolites rather than the parent compound, that is the toxicant. And we have the third case that is the most challenging one, and that is the one with general low or no toxicity, because you don't know really what you are looking for. And then we have the fourth case that is really like you have actually structural similar compounds, but you can have sort of a hypothesis what are the different mode of actions, and they might sort of subcategorize to different mode of actions. So we had that first workshop in 2014. We met, and from that point in time, we have been working on the different case studies under the different scenarios. There was also this publication and a strategy how to make and report a read-across prediction. And we met again last week to see where we are, and that is the photo is from last week. And all this will feed in to the future, of course, because of course we know this, what Thomas said before, that we are sort of always stressed about our projects that are finishing after 4 years, 5 years, and so on. But we are sort of trying to feed in the lessons learned and the output from the SURAT project into the future. And this is a topical scientific workshop that will be at the ECHA is organizing in April next year, and we will contribute with a couple of our case studies to this workshop. And this is of course also to see then the application of the read-across assessment framework, and also see how the new methods can be used in safety assessment. And then we have this final event that is coming up very soon. It's the 4th of December in Brussels. We are now desperately trying to collect everything we did to make it presentable at this workshop. And it will be a nice event. Everyone is welcome. Everyone can come and register. And we will have some sort of speeches in the morning, providing an overview of both Surat 1, what it have produced, but also the context in the world, relation to similar projects, and also looking forward in the future. And then in the afternoon, we have actually an exposure session where you can go and look at the different models and methods that were developed within the Surat project, and also a poster session. We also provided this paper that is also providing the lessons learned to give some idea what could be the future research that could be based on what Surat did. And of course, we are also feeding into the EU Tox Risk 21 project that Thomas mentioned. And this project is basically taking over some of the parts or the results of Surat 1, and that it will do it is quite guaranteed that it's a big overlap in between the partners of the two projects as well. So I don't believe that we are going to lose what Surat 1 did manage to achieve during these years. So thank you for the attention.